Hey there, welcome to EduDose, your trusted source for pharmaceutical and microbiological learning. I'm here to walk you through one of the essential foundations of microbiology, culture media, their types, and how we maintain them in labs. So, what are you going to learn today? By the end, you'll understand the purpose of culture media. Tell apart different types like selective, differential, and enriched media. Know how we keep cultures alive in the lab, and see how all this connects to real-world examples. Let's start simple. What is culture media? Think of it as a nutrient-rich substance that helps us grow, isolate, identify, and study microorganisms in the lab. It supplies carbon, nitrogen, vitamins, and all those essential ions microbes crave. Culture media comes in two main forms: liquid like nutrient broth and solid like nutrient agar. Now let's break down the types. First up, enriched media. This is used to grow fastidious organisms, basically microbes with really picky nutritional needs. For example, blood agar supports the growth of Streptococcus species, while chocolate agar is perfect for Neisseria gonorrhea. Where does this matter? Diagnosing respiratory or sexually transmitted infections. Next, selective media. This kind of media encourages only certain microbes to grow while keeping others out. McConkey agar, for example, selects for gram-negative bacteria, and mannitol salt agar is great for Staphylococcus species. We use these for things like detecting foodborne pathogens, water testing, or clinical samples. Now, differential media. Here's where things get interesting. Differential media helps us tell microbes apart based on their biochemical reactions, like sugar fermentation or hemolysis. Take McConkey agar again. It differentiates between lactose fermenters, which turn pink, and non-fermenters. Blood agar shows different types of hemolysis: alpha, beta, or gamma. This is super useful for identifying enteric bacteria and streptococci. General-purpose media is up next. It's as straightforward as it sounds. Just supports the growth of non-fastidious organisms. Think nutrient agar or tryptic soy agar. Perfect for basic growth, maintenance, and subculturing in the lab. Don't forget transport media. These special media help preserve specimens during transport, preventing overgrowth or drying out. Stewart's medium, for example, is used for Neisseria species, and Carey-Blair medium is great for stool samples. Super important for clinical diagnostics and field surveys. So, how do we keep these cultures happy and uncontaminated? Maintenance is key. Periodic subculturing onto fresh media, refrigeration at 2-8 degrees Celsius for short-term storage, deep freezing at -70 degrees or below with cryoprotectants like glycerol, and even lyophilization or freeze-drying for long-term storage. You'll need slants, screw-capped tubes, cryovials, and a good laminar airflow hood. Why does all this matter? Applications are everywhere. Antibiotic testing, pathogen identification, vaccine production, environmental monitoring in sterile areas, and the fermentation industry. Let's recap real quick. Enriched media, extra nutrients like blood agar. Selective media selects one group like McConkey. Differential media shows differences like hemolysis. General media for basic growth like nutrient agar. Remember, a good culture medium is not just a nutrient bed, it's a diagnostic canvas that reveals the hidden world of microbes. If you found this video helpful, make sure to like, share, and subscribe to EduDose for more on microbiology and pharmaceutical sciences. Don't forget to hit the bell icon for updates. Thanks for tuning in, and see you in the next lesson.
If you found this video helpful, please subscribe, like, and share it with others who'd benefit. See you.